how it is actually more like in reality, not following these like diagrams. All right, so everyone, over here, over here. It's okay, relax, we got this, right?
Just dropped like a bomb on us.
What happened there? What happened there? What's going on? Everything good?
Yeah.
I feel some panic attacks, anxiety.
Do you drop the lowest grade?
No, but the exam can replace.
Look.
The exam can replace your lowest grade.
The final exam?
One exam can replace your lowest grade.
Can you replace like all of them?
A couple of them?
Two of them?
No. Alright. Let's discuss that more after your exam, right? Right now, do not make plans. What am I going to do? No, that's a failing
I'm already
mindset.
enrolling in your next class.
What's that?
Like I'm already enrolling in your next class.
Okay. You know, come on, you have to believe in yourself. You got this, alright?
You're trying to study is hard.
Okay, so if what has happened, it hasn't changed, then
Try
let's
something
make
new.
some changes, right?
We did.
Exercise sessions. Okay, exercise sessions.
I don't have time for that.
Well,
Oh yeah.
Hey, you can try night owl tutoring. I'm the CEO.
I'm a future accountant on that.
all right. So let's go back together in class. If you need some help, there is office hours, there is exercise sessions, there are some opportunities for tutoring. The natural science department has some tutors. You can use those as well to help you with your classes. Okay, so now we go for the proofreading and preparing DNA. Like I mentioned before, these DNA polymerase, they are really fast and really accurate, but still, there are some errors that takes place, okay? There are some, for example, some incorrect nucleotides that can be added there. So this mismatch repair of DNA include these other enzymes that will basically correct these errors in this base pattern. So for example, a hereditary defect in one such an enzyme is associated with a form of colon cancer. Once you have like these mutations, or if they get like transferred or they get passed on from one generation to the other, then associated, for example, with this colon cancer. And this defect allows for these cancer-causing errors to accumulate in DNA faster than normal. So that's why you have this mismatch repair of DNA. This DNA can be damaged by exposure to harmful chemical or physical agents, such as, for example, cigarette smoke, X-rays, UV light. It can also undergo like spontaneous changes. So in this nucleotide excision repair, or this nuclease cuts out and replaces the DNA damage and stretches this DNA, so in order to be repaired. Okay, so this nucleotide excision repair. This mutagenesis basically can be occurred, can happen by this industrial chemical or natural carcinogens, or even, for example, in endogenous sources like inside the cell, depurination, free radicals, DNA polymerase errors, and then you have this DNA repair. If it is not repaired, then this unrepaired DNA damage or these miscombinated nucleotides, then those can lead to these passive mutations from one generation to the other ones, and those are the ones that will cause, like, for example, this cancer and so forth. So here's basically the example of this mismatch repair. Here you have the incorrect nucleotides right there. They're not supposed to be there. They were added there in error. They were caused there by exposure to different chemicals and so forth. In order to repair that, here you have this nuclease, this enzyme, that will cut this DNA where you have the error. It will cut it, and then DNA polymerase 3 will come and it will replace this DNA that is wrong now with the correct one. Okay, so you have first the enzyme cuts, then you have the DNA polymerase, it will elongate the correct, and then you have the correct nucleotides. Then you have the DNA ligase that will come and join at the end of these DNA segments. So make sure you know these key players. Know the key players for DNA replication, and know the key players for the DNA mismatch repair. Okay? So this error rate after this proofreading repair is low, but not zero. Right? So there is still some errors, some errors. And these sequence changes may become permanent and can be passed on to the next generation if they are not returned, they can be passed on. And these mutations, these changes, are the result of this genetic variation from which natural selection operates. Okay? So if it is harmful, then there will be deletility risk. Yes, question.
Can you go back? What happens if that middle thing breaks?
What middle thing?
On the second one?
Right here?
Yeah.
There
Like,
it broke.
what does it cost? Like, it's just. What does it cost? Anything like in general?
What is the cost?
Yeah.
Oh, so that's the mutation right there. If this mutation, that is this one right here, if this mutation doesn't get repaired, and we will see later on the next module like what type of mutations there are. If this is a harmful mutation and it doesn't get repaired, then that's what it's going to cause like different diseases. Like for example, the one that we will see in the lab, sickle cell disease. The example that I gave you for colon cancer and so on. So if it is a harmful mutation, then it will cause disease and it can be passed on to the future generations. So that's why you have this mismatch repair. Break it, replace it, join them. Okay? Good question. All right, so yes. So these limitations of DNA polymerase create these problems for the linear DNA of eukaryotic chromosomes. This is what I mentioned before. Remember when I told you, keep this in mind for these linear chromosomes, how at the end you will have like primers. Okay, so what do you do with them? So these linear chromosomes, they have these limitations whenever they're going to get replicated. So the usual replication machinery cannot complete these 5' ends on the daughter strands. So this repeated runs of replication will produce these shorter DNA molecules with uneven ends. How is that possible? So that's basically what you have here. Remember when I showed you this? Picture this in mind, but this strand, that is the template, and then the new strand that is created, same thing over here. Well, notice here, this is the RNA primer, okay? This cannot be replaced, why? Because it will need to have sequence on the other end to replace that RNA with DNA, and then DNA polymerase 3 to come and join and elongate and then ligate, right? So you cannot do that at the end. So what is going to happen? So here, this one is going to get replaced, is going to get joined, but here you will not have that replacement right there, okay? So now you will have uneven strands right here, shorter DNA. What happens with that uneven strands right there? So you begin here, right? And then second round replication, you will only replicate this right there. And it will continue to do that for the next round of replication, shorter DNA, every single round of replication. Okay? For cells that divide faster, obviously this will create a problem, right? So what do you do there? So in these eukaryotic chromosomal DNA molecules, they have this special nucleotide sequence at the end that are called telomeres. These telomeres, they are these repeat sections of non-coding DNA that were added at the end of these linear chromosomes.
Placeholders?
Hmm?
Placeholders?
You can call it that, yes, like this, on this end, the telomeres will be at the end.
Okay.
These repeat sections of the non-coding DNA.
But they're like placeholders?
Hmm. Yeah,
Yeah, okay.
but this one at the end,
Okay.
right? You can call them like placeholder, but these are like telomeres.
Okay.
There will be like these repeat sections of non-coding DNA, like that. And so they do not postpone this erosion of DNA, right? They do postpone it, but they do not prevent it. They do not prevent it, this shortening of DNA, but they postpone it until it gets to the coding DNA. So remember this. This is the coding DNA, right? That's where you have your genes, linear chromosome. So you add them at the end, and that is your telomere sequence right there. Okay, that's what you have there. This telomere sequence, these sections of DNA, they are non-coding DNA. So in other words, you don't have any genes right there, right? So you can have this as a buffer, okay?
I thought that had to be live.
Hmm?
So it's supposed, the pink one's supposed to match the orange, right? No, but I thought it would have to be like coming the other way, kind of like
Like in
the
the
IRAP.
Well, there you are thinking about the whole strand replication of how one matches the other one,
So
right?
what is this?
This one, don't worry about the color. This color is just to tell you that it is the extension of this linear chromosome right here. That one, where you have to worry about like the matching, that's on this region of DNA. Okay? That's where you have your coding DNA. That's where you have your DNA. That's where you have your genes. Okay? No, no one. No one.
I don't know what you're trying to tell me.
Huh?
I don't get it.
Okay, so you were worrying about the different colors, right? Like this one.
No, I'm not worried about the different colors. I'm saying like, so you're saying the pink one's on top and we're supposed to match the orange one with what's on top. I thought it was supposed to be like the IRAC where it's supposed to be in,
So is you
Anti-parallel, right? Yeah, yeah, but not
no.
necessarily this. Here the G is can only bind to the C's, right? The T is can only bind to the A's, right? So it is still the same.
Can I clarify something?
yeah.
Okay, so on the IRAT, it was a little bit different because first you had to translate it, which right there, that's translated.
Uh-huh.
Then it was asking for the opposite thing. So it would be one more step on the IRAT. It would be that, but then you would read the bottom one backwards.
You're talking about the direction, 5' to 3', right? So again, that's what I mentioned at the beginning, right? That's the coding DNA, 5' to 3'. Then the opposite one will be just the anti-parallel, but going from the 5' to 3', right? So whenever I ask you for that, that will be for the DNA sequence, well, assuming that it's going to be like coding DNA, and you have to give it from the 5' to the 3'. Okay. These are telomeres. I will not ask you for that on the telomeres. Right? Telomeres, I will ask you what are they and what are they for. So in other words, here you will tell me this telomere sequence. These are complete sections of non-coding DNA. That is keyword, non-coding DNA. In other words, they don't have any genes in there. And these repeat sections of this non-coding DNA serve to place at the end in order to prevent this erosion as DNA replicates. So it will not create shorter DNA that will damage your coding region of
The
DNA.
one that prevents short DNA.
What was that?
They prevent short DNA.
They prevent short DNA on your coding DNA, your coding region, the one that it has your genes. Okay? Yes.
Will the telomere be the same, like for all your cells? Like, will it be the same sequence?
Yes, those are the repeat sections right there.
But for like all the DNA strands at the end, will it always be
For the
same?
chromosomes, yes, yes. It will be at the end, yeah. So, those could be like placeholders, those could be like known as buffers, right? Buffer in the sense that, they will serve to prevent these shorter regions on your coding DNA. This is important stuff, this DNA. Once this shortening of DNA comes, like let's say it finishes all your telomeres, and now it goes into your coding region, then that's when you have problems. Right? Think of, for example, Alzheimer's. Right? This erosion of this DNA is more like seen like in the aging. Okay, so it will erode your coding DNA. It will get into your genes. It will get into the coding DNA region. Those genes, then you will not have those genes. Then that's going to create problems. Okay, so telomeres, what are they? Repeat sections of non-coding DNA serve as the end of the chromosomes to prevent the erosion of your coding DNA. Okay. They postpone it, yes, in every single round replication, you are going to get reduced on the telomere sequence length, right? So in every single round replication, you lose some telomeres, you lose some telomeres, you lose some telomeres. But the whole point of that, with the telomeres, is to do that. So you will lose that type of DNA, and you will not lose the coding DNA. Okay?
Is it coded or what?
Coding, like
Coding.
C-O-D-I-N-G, coding. Coding basically means that it has, it codes for genes. Okay? Because from that, you will have the genes, right? Then you have transcription, make it RNA. You have translation, make it into a protein. Okay? So that's what you need it for, this particular coding region. So the example that you were mentioning, the one that I have on the IRAD, I have this sequence right here, 5' to 3', and then I ask you, what is the sequence right here? So you have to tell me 5' to 3', the sequence that it will bind to, right? The anti-parallel sequence. Here, I will not ask you for the sequence of the telomeres. But just know that it's the same thing, right? This one will go, let's say, 5' to 3', This one is 5' to 3'. So basically the same thing. It's just the direction that I'm asking for. Okay? Any question on this one?
I have a question.
Yeah.
On the bottom thing, is that saying that it's shortened? So
Yeah,
like,
so-
okay, so that's the telomere sequences, right, on the end, the bottom?
Yeah, yeah, right there.
Yeah.
That's like short.
So is it just like showing you that like it still shortens but not as much?
It would shorten in every single round.
But it doesn't prevent it?
It doesn't prevent
Is that what that diagram
yeah,
is showing?
Yeah, it doesn't prevent it. You will still lose DNA. It doesn't prevent it, but it just
It slows it down.
postpone it until it gets to the actual DNA, to
The coding DNA?
Yeah,
Okay.
yeah. So it loses DNA in every single round, but the whole point for that is to serve as a buffer, so we will not get your genes from the
Mhm.
coding DNA.
So it's like a cap?
It's like a cap, yes, like
Okay.
a cap. Yeah, put there like a cap, and the whole cap it is to, so it will not get to your important DNA.
Okay.
Okay. And once it gets there, and if you lose DNA or important DNA, then that's what can create problems, right? Alzheimer's, aging, think about like, yeah, the longer that you live, the more non-replication and like that. That we see there, those telomeres at the end of the chromosomes, right there.
I thought they just finished mapping the entire genome and they're still trying to figure out what telomere means, like in March.
In March.
Yeah, they just finished mapping telomeres, which was like 8%.
Send to me to see like what are like meaning with that. I mean, probably like
But
for.
they don't really understand what it does.
Well, we know these parts, but there has, with many things more in biology, there are like many other functions. that
They don't
play.
fully know,
Hmm?
right? It's not, it's, they know, but they don't fully know.
They fully know, yeah,
Yeah.
yeah. Think of, for example, the liver, right? There are like tons of different things that the liver does, so like finding out. Just, yeah, please send it to me to see like what they're like meaning. So, here, if chromosomes of germ cells became like shorter in every cell cycle, then these essential genes would eventually be missing from the gametes they produce. So that will function with telomeres, right, to prevent this. That's what they do. I think I know. Yes, you know, before, right? You will not only for like telomeres, but also like about like junk DNA, right? Many junk DNA, and they just compare the coding DNA. There was like many more junk DNA. Now we're like trying to, well, like finding out that it's actually not junk DNA, so you can actually see all the functions. So yes, like, we keep finding out. That's what I mentioned. Like we are standing in these shoulders of giants. We keep increasing this knowledge gap by many of these more scientists and their discoveries. Right. So that's why it's important. And this enzyme is called telomerase. That is the one that catalyzes the lengthening of the telomeres in germ cells. So that's what is important. And this telomerase, this enzyme, has been shown that it's active in cancer cells. So this telomerase is not active in most human somatic cells because it will add the telomeres already. But in cancer cells, it will keep adding. So it is active, right? Because cancer cells, they will be dividing like crazy. And if they are dividing like crazy, they are doing lots of DNA replication. And doing lots of DNA replication, they will be losing lots of DNA. So in order to prevent that, to get into the important genes, telomerase is active in order to keep adding those telomeres at the end. So telomerase is currently under study as a target for like cancer therapies, like block it. So cancer cells will not be using this enzyme, they will not be adding telomeres, and by blocking this enzyme, so they will start losing DNA as they are replicating, kind of like killing them on their own, right? By their own choosing, because they decide to divide rapidly. They don't have this guy to help them protect against the shortening DNA in every single round replication, so they will die. So that's basically what they're calling this. Okay. Any questions, comments?
I have a question.
Yes.
What about stop codons?
The stop codons, yes, that will be for the translation. Once you have the gene expression, like transcription translation, the stop codon will basically tell you like where to stop for the translation process.
And
So-
that's not in cancer.
Hmm?
Like, why can't they do that? Why can't they artificially make a stop codon for cancer?
They could, for example, in key players involved in cancer, right? That would be like one way to target that. Let's say, for example, you know, particular cancer, it has this particular mutation or it expresses particular protein, high expression. then adding a stop code on that would be a key, right? But for that, you will have to find out, okay, which type of cancer it is, right? And how will you introduce that to which type of cells? Where will you introduce it? So it's more like specialized therapy. Okay.